When talking about genetics, may it be microbial or basic genetics, the first thing that comes into our mind is probably either chromosomes or DNA. But if you think about it, isn't it amazing that through the years of understanding science, we've come this far, down to the littlest things such as that? Haven't you wondered what enabled us to study these materials in the first place? Well, I might just have the right answer, and it all started with the extraction and isolation of the DNA. We might already be familiar with the basics of DNA, its structure, the information it holds, the transcription and translation process, and stuff like that. But what we might not be acquainted well with is the first step that enabled the study of these things. Extraction of DNA is the initial process that we go through in experiments and studies that deal with genetics. And of course, there are various methods to do this. The most common methods include the phenylchloroform method and the proteinase K method. In laboratories, though, to save time and ensure the quality of extracted genetic materials, they often go for kits that employ the spin column method. But the basics of any DNA extraction are just the same. Typically, the general steps followed are like this. First, we introduce a sample, be it of a plant, animal, fungi, bacteria, or any other living organism, and mechanically break down its first layer of protection, quite specifically among fruits and leaves, for they have this cell wall. Sometimes, though, we also centrifuge it for small samples suspended in buffers, which were initially found in cultures, right? Then, the next step for this is that we add a lysis buffer into our sample at a controlled pH level, typically at around 7 or 8, because this process of DNA extraction can be quite a pH-sensitive process. This buffer then contains SDS, or sodium dodecyl sulfate, along with a chelating agent like ethylene diamine, tetraacetic acid, or EDTA. Sodium compounds such as sodium chloride or salt are also added to help remove those pesky proteins bound to the DNA. If not, proteinase K is added. In the end, the main goal of this step is to lyse and break open the plasma membrane of the cell and release its contents like DNA into our solution. After the lysis step, we move to the purification method that usually uses sodium acetate for the negation of the DNA charge and chilled ethanol or isopropyl to precipitate our DNA. Sometimes we even go an extra step by adding RNase to ensure that our solution is solely populated by DNA alone. By this point, we have extracted our subject. The other steps from there on includes the washing and further purification, depending on the method you are following. At the end of it all, once purified, DNA is stored in a stable buffer like tris EDTA and may now be used for experiments. So now that we know the general steps, the question is, can we perform this by ourselves? Well, absolutely, and today that is exactly what we are going to do using common household materials that I have here in front of me as alternatives for all those solutions that I have mentioned earlier. So, let's get it started! Right around here, we have a few materials for our experiment. As we go through the steps, I will be explaining the rationale behind each of these materials. First, take a handful of your leaf samples and put it in the blender along with about 100 ml of cold distilled water and a half teaspoon of table salt. Cover the glass and start blending. The reason why we are blending our plant material is so that we can break down its cell walls. As we know, plants do have quite a tough exterior wall before we can even encounter the plasma membrane that is typically the first layer in other organisms. On another note, the addition of salt earlier was so that we could initially remove the proteins from the cell and DNA by dissolving them in the aqueous layer. The sodium ions of sodium chloride becomes temporarily attracted to the negative phosphate backbone of the DNA, neutralizing it and making it hydrophobic. Next step into our extraction is to filter our solution through a strainer. This is done to remove the physical debris that resulted from blending earlier. Then, after filtering, we will now proceed to the lysis step of our DNA extraction. We will be adding detergent, or in this case, dishwashing liquid, into our solution. The dishwashing liquid acts as the sodium dodecyl sulfate for the general step of DNA extraction, and it dissolves the cell membrane by denaturing the proteins and lipids in the layer. By lysing the cells, the contents of the DNA spills out in the open. To ensure that we have sufficiently lysed the cell, we keep it untouched for about 5 to 10 minutes. After waiting, transfer the solution into a different container. Then to further purify the DNA we extracted, add a few grams of meat tenderizer. Common ingredients of meat tenderizers include the papain enzyme, a protease commonly found in papaya fruit. This enzyme helps in the digestion of potential proteins that are still hanging around and bound to the DNA in our solution. As I mentioned earlier, proteinase K performs this purification method in traditional laboratory extractions. To ascertain that the digestion reaction occurred, gently agitate the container to mix the solution adequately. Now, we will be isolating the DNA itself from the solution. To do this, grab your chilled isopropyl alcohol or ethyl alcohol and gently add it into the mixture. Wait for about 5 minutes for the separation to be completed. As we wait, let us discuss a few things about this step. The reason why we add alcohol is because it is a non-polar solvent. The solution we have is aqueous and polar, so the hydrophobic DNA that resulted from the sodium phosphate binding earlier will become even more hydrophobic and will start to move towards the non-polar region in the solution. As for the temperature of the alcohol, which is low due to being chilled, it is a precautionary step as DNA may denature in hot temperature. After waiting for a few minutes, here is our extracted DNA. It appears as clumpy and stringy white material in the alcohol layer of the solution. Typically in the laboratory, this will be stored in tris edta buffer to ensure that it is in stable pH and will be usable for future experiments. So, there we have it. It is true that we can extract DNA from plants using common household materials as long as we keep in mind the general mechanism of the extraction method. This is generally the same idea in other organisms, even in plasmid DNA, which is a little bit different than genomic DNA. Plasmids are circular and supercoiled compared to genomic DNA of linear presentation. Due to this, it requires a little bit more complicated steps in order to extract, but the idea remains the same. In the alkaline lysis method of extracting plasmid DNA, we use 3 solutions of varying compositions. The first solution contains glucose and tris edta to keep the samples in a stable pH as we go through cell lysis, initiated by the second solution that also has SDS. The only difference is that this time around, we also add alkali sodium hydroxide in order to break the hydrogen bonding between genomic DNA. Remember, we are extracting plasmid DNA this time around, and due to its circular orientation, it will not be affected by the sodium hydroxide. Then the next steps after cell lysis is still the same, the isolation and purification. The third solution, with acetic acid to drop the pH, is added to aggregate the genomic DNA. Along with this, potassium acetate is used to remove the SDS molecules so that when we centrifuge it, this debris will be left in the pellet and the supernatant will have the plasmid DNA. Isopropyl is still used as a precipitating agent to clearly separate the DNA that we have extracted. So overall, the steps are still quite the same, only that this time we have extracted plasmid DNA instead of the genomic ones. These plasmid DNAs are of great importance in the laboratory as they are usually experimented on to be vectors in studying cures and even antimicrobial resistance among pathogens. As a recap, in this video we talked about the different methods of DNA extraction, the general steps involved, the at-home alternatives of solutions for DNA extraction at home, and of course, the plasmid DNA and its extraction. Thank you very much for watching, and I hope that you picked up something worthwhile. Bye!